All right. Good afternoon, everybody. Thanks for coming today. So it's my great pleasure to introduce Jordan Horowitz today. He's visiting us from the University of Michigan. As you can see, he likes to collect affiliations with departments. He's in physics, biophysics, and complex systems. And he truly bridges all those communities. And you'll see that today. Just to give you some context, he did his PhD in physics with Chris Darczynski, who some of you that are Nonequilibrium aficionados might recognize. And then he did a bunch of cool things. As a postdoc, one of them, he was a living systems fellow, physics of living systems fellow at MIT with Nikta Fakhri, who gave a colloquium not too long ago with Jeremy England and with Meran Kardar. And for some of that work, he actually received the Erwin Oppenheimer Award from the Physical Reviews for the best paper from a young investigator. He also has other accolades such as the NSF career. And, he really works at this intersection with physics, biophysics, complex systems, as you see from all the titles. So I'm just repeating the titles of the departments. But thinking a lot about the theory and the practical aspects of non-equilibrium thermodynamics, non-equilibrium statistical mechanics. And I think it's very cool to have him here. It's like, this is mostly, people that know this are mostly my fellow faculty members, but we had a review for the department. This happens every 10 years, and as part of that, as a faculty, we got together, and every week somebody would give a talk about one of the great new challenges that the department could try to solve that could face in the next 10 years. And we got free lunch, so that's always good. And somebody gave a talk, and one of the ones that I had to put together was the one on non-equilibrium statistical mechanics. And coming from the side of biophysics, you can see that there's lots of examples where biology is pushing our understanding of non-equilibrium physics. But one of the interesting things was that as we interviewed the rest of my colleagues, it was clear that in most fields or subfields of physics, having an understanding that goes beyond equilibrium thinking could be of use. So I think it's a really, really interesting to have him here to talk about this, and also to give you some context. He's here for the whole year as a visiting professor with the Miller Institute. He's actually hanging out in my lab, so he's got plenty of time to talk to you. And on a more colorful note, it's been really fun to hang out with a hardcore theorist every day, because every time he says he's getting upset because every time he says, why can't we do this? And I tell him, well, the data is coming up. It's coming up in, I don't know when. So it's been great. And I think you're going to see how he is at the boundary of non-equilibrium thinking and non-equilibrium physics, but also in a very pedagogical and accessible way. So again, he's here for the whole year until the end of the academic year. So I encourage you to contact him if you want to hang out. So with that, please join me in welcoming Jordan Horowitz.
Okay, thanks, Hernan. That was a very kind introduction. It was very nice. You kind of stole my intro, actually. I'm excited to be here to tell you all a bit about what we've been doing in the group, and really trying to think about how do we create quantitative limits to non-equilibrium phenomenology? How do we build out this kind of modern non-equilibrium framework to really say something new and interesting? And in kind of thinking about what a non-equilibrium framework or non-equilibrium prediction would be like, I want to take a step back and remind ourselves about what kind of predictions we get from equilibrium thermodynamics to get a sense of what we can hope for in a non-equilibrium thermodynamics. And so for that, I want to take you back to your first year physics class with the efficiency of a heat engine. Here on the left is a picture of a classic steam engine, right? This is like everything we've studied in our first year. So to remind you, a heat engine is a cyclically operating device. It operates between a hot temperature reservoir at TH and a cold temperature reservoir at TC. And the idea is to operate in a cycle and pull energy from the hot reservoir, pull some of that energy out as work, and deposit the rest of it as heat into the cold reservoir. And one of the central quantities of interest in thermodynamics is the thermodynamic efficiency, which is the ratio of what we get out to what we put in. And so we all remember that the second law of thermodynamics tells us that the efficiency is always bounded by the Carnot efficiency, which in this case is 1 minus the ratio of that cold temperature to the hot temperature. Right? So what is this? This is a universal constraint on what's possible. Okay? It tells us that no matter how I build my heat engine, no matter what materials I use, the best I can do is, as long as there's a hot reservoir and a cold reservoir, is given by this efficiency measure, right? If I want to make, it doesn't make specific predictions about what the efficiency of a particular heat engine is, right? It doesn't tell me if I build my heat engine out of rubber, or I use oil, or I use fuel, or I use water, what the efficiency will be. I need another theoretical framework to do that. I need to pull from material science and fluid dynamics to make those predictions. But what thermodynamics does tell us is that no matter how you decide to build it, there's a best that you can do. Moreover, if we ask when do we get an equality, when do we saturate this inequality, it gives us optimal design principles. How should we build the best heat engine? And we know from the second law of thermodynamics that what we should do is operate very, very slowly. Okay? So we can learn how to do the best once we have this kind of bound. But as we all know, that much of nature is out of equilibrium, and we would like to make similar kinds of predictions. As we drive a system out of equilibrium and start consuming energy, can we get something out of that energy, and what can we get, and can we make that a quantitative statement? So up on the left here is an image from my colleague Nikta Fakhri's lab. What you're seeing here is the expression of a protein just along the cell membrane. You can see it forms this dramatic dynamic pattern. And one of the questions you might be interested in asking is, how much energy does it take to create a pattern like that? If I put more energy in the system, can I create more precise, faster patterns or better patterns? We also know that information processing requires energy. We know this because we have to plug our computer in, and then it gets hot because it's dissipating energy. So a natural question is, if I use energy, can I process information more reliably or faster? Subcellular systems like molecular motors or chemical clocks are functioning inside the cell, and they're using energy. Can you create function better, overcome fluctuations? And lastly, I'll note this video in the bottom left. This is a classic soft matter experiment on active matter. What you're looking at here are micron-sized plastic beads in a fluid that have been chemically coated to kind of zip around in a particular direction. You can see they form these clusterings and structures. And of course, the question then becomes, if I'm putting energy into a system, how does that relate to what kind of clusters or structures that I can form? So inspired by equilibrium thermodynamics, we're going to start asking questions about, if I have some function, pattern formation, information processing, right, energy extraction, as I drive my system out of equilibrium, I should be able to do better at that function, okay, and quantify that through inequalities. So when you can imagine that you have some measure of your efficiency or efficacy, how good your pattern is, and you're driving out of equilibrium, and you can think about this space of possibilities, that for any particular system, as you drive it out of equilibrium, you'll be able to do your function sometimes better, sometimes worse, just like in equilibrium thermodynamics. Right? Sometimes you make a great heat engine, sometimes you make a bad heat engine. But whatever there is, there's a best heat engine, a best function that should increase as I go out of equilibrium. And our goal would be to try to understand, what are these limits? Right? What is the best we can do out of equilibrium to understand how we can create function by driving a system out of equilibrium? So in the last few years in this space, we've been working on a few different kinds of projects. The first is really about making processes more precise. So if you have a non-equilibrium process that you want, like a clock, and you want it to be very precise, you might imagine that if I dissipate energy, I can increase precision. Can we make that quantitative? Our focus of late really has been on this idea of driving systems out of equilibrium to making them more sensitive. So if you perturb a system by changing some input and look at an output, as you drive the system out of equilibrium, can you make it more sensitive? How much? And then once you're kind of interested in non-equilibrium systems, you can start asking questions like, what are the time scales of dissipation? Can we make quantitative statements about that? And over the years, we've also been generically interested in non-equilibrium fluctuations and energy consumption. What are the energetic limits to suppressing fluctuations, the energetic limits to processing information? We can do all these kinds of things using modern tools of non-equilibrium thermodynamics. But today, I want to focus on this first two, the energetic requirements of suppressing fluctuations or precision, and the energetic requirements for driving to creating very sensitive, responsive non-equilibrium systems. So before jumping in, I want to be sure I do the acknowledgements. A lot of what I'll be talking about today got started with Todd Gingrich, who's now a faculty at Northwestern. Much of the work was also carried out Jeremy Owen, who's a postdoc at Princeton, as well as Sean Fancher, who's a postdoc in my lab at the moment. Okay, so I'm going to drive a system out of equilibrium, and I have this idea that I can make it more precise by burning more energy. What can we say about that? So I'm going to try to motivate this with a molecular scale clock. So there's bacteria, cyanobacteria, so molecular scale clock. The idea here is that we have some molecule. It's going to go through a series of configurations to kind of keep time. And this example is drawn from cyanobacteria. So cyanobacteria live in ponds, basically. And they harvest light for energy. And so they have to know what time of day it is. They have to rise to the surface to grab energy, and then they sink back down. And so they have to find some molecular mechanism, because cells are just bags of chemicals. They have to find some molecular mechanism to keep time. Okay, and so a simplified version of this is the idea that you have some molecule. It goes through a series of molecular configuration changes. And then when it returns back to its irregular configuration, that's like 1 clock time. Okay, that's how it tries to keep time. So we could imagine an experiment where we're following the system going through these sequences of configurations. And at every moment in time, we put a little dot when we see that cycle completion, right, at 1 clock time. Now this is a molecular scale system, right? There are fluctuations, there are thermal fluctuations, and the time between those completions is going to be random. Sometimes it's going to be a little wider, sometimes it's going to be a little tighter, okay? So one might imagine that if I drive the system out of equilibrium, if I put energy into this system, that I might be able to make that sequence of dots more regular and therefore create a precise clock. Because that's what you want a clock to be, right? You don't want your clock to speed up and slow down randomly. You want a second to be a second for a long period of time, okay? So what we found is that instead of quite studying this problem, it's analytically and theoretically convenient to imagine that we fix an observation time, okay, and count the number of events that happen in the observation time, okay? So if the number of events, if the time between events, little dots is fluctuating, then the number of events in a fixed observation time will be fluctuating as well. And if the number of dots are very evenly spaced, then the number of dots in a fixed observation time will be fixed as well. So that's what we're going to study. And to kind of get concrete, I want to draw another biological example because we actually see this cycling in person. And this is from the F1 F0 ATPase motor. This is a molecular motor. It sits in the membrane of mitochondria. And its job is really to from one form of chemical energy into another form of chemical energy. And in fact, it makes adenosine triphosphate, or ATP, which is a molecule used in cell kind of like its battery, its energy currency. So it makes the ATP, the energy currency that goes off and creates, powers other processes. And what's really amazing about this machine is it does that through mechanical motion. It turns chemical energy into chemical energy through mechanical motion by actually physically rotating. And so a classic experiment is in the bottom right corner here. We're actually seeing that rotation. What was done is that basically a rigid polymer was attached to the top of the motor. Energy was supplied, and you can see it starts to rotate. It starts to spin, but it's quite noisy. And the question we're interested in is what are the trade-offs with supplying energy to the system to make it spin very precisely? So here, energy is coming from the system. Here the system is actually running backwards. Energy is coming from the addition of ATP, the addition of these high energy molecules. And the cost is the heat dissipated, which in this case is just how much ATP per unit time you use times the chemical driving force in this particular case, right? So classical force times flux relationship. So as we burn more ATP, can we make this thing more precise? So let's think about that. Let's imagine a hypothetical experiment where we're watching this thing spin around, and we have a little counter in our hand, okay? And every time the thing spins around in a clockwise direction, I'm going to add 1 to the counter. And every time it spins around in the counterclockwise direction, I'm going to subtract 1 from the counter. And then I can plot the value on my counter over time, right? Over time, it's going to grow on average, but there clearly are fluctuations. And after some fixed observation time, I'll have some number, J of t. I'm going to call that the integrated current, okay? So everything I'm going to tell you about today isn't just for spinning molecules. It's any integrated current, anything that builds up over time. So it could be the distance a Brownian particle moves in some finite time, the distance a molecular motor transports, or the number of electrons that move through your resistor, okay? So we have, in this case, a molecular scale system, a small system. There are thermal fluctuations, which cause this curve to fluctuate a bit. But moreover, if I were to repeat this experiment many, many times, I get many, many different traces, right? Clearly, there's noise, and their fluctuations are spreading things out, but there's also clear some average growth, some average current. What we'd like to understand is the size of those fluctuations over time and compare them to this average current. So what we're going to do is we're going to imagine making vertical slices through this picture. And at each time, an early time blue, a middle time orange, and a late time green, we're going to histogram our measured values of current and try to look at the fluctuations. So if we look at the early time in blue, and I histogram all the values that's plotted on the right in this blue curve, you can see that the curve is on the left. There hasn't been a lot of time for current to develop, and it's fairly skinny. There hasn't been a lot of time for fluctuations to develop. But over time, as we look at later times, our histogram of value shifts to the right and starts to spread out. Right? So over time, the current is starting to develop, so the distribution moves to the right, and there's been more time for fluctuations to develop, and this distribution is spread out. Okay? And so what we're going to do is use that spread, which is capturing the magnitude of fluctuations, to measure how much noise there is in the system, so the variance. And then the question is, how do we reduce the fluctuations in this current relative to the mean? And what we've shown is that for pretty much any non-equilibrium system, there's a fundamental universal trade-off between the precision or relative uncertainty, the ratio of these fluctuations to the mean squared, against the energy dissipation. That the relative uncertainty is always bigger than 2 over the observation time times the heat dissipation in the steady state. So I often like to think of this as kind of like a Heisenberg uncertainty relation kind of thing, right? So if I want to have really low fluctuations, I have to pump energy into the system. But if I'm willing to allow large fluctuations, I can do that with less energy. So this result was originally proposed, hypothesized by Andre Barato and Udo Seifert. Our contribution here was to prove this for any steady state system that can be modeled as a discrete state Markov process or an overdamped Langevin equation. Okay. So we have a theorem and a proof. I can tell you a little bit about it, right? We can analyze what are the best systems, what are the optimal systems. You are closest to the bound when the fluctuations are Gaussian or near equilibrium. Moreover, I said this works for any integrated current, anything that builds up in your system. Heat itself is something that builds up in your system. Heat dissipation is something that occurs over time, that's building up over time. And of all the currents that you could look at, the one that comes closest to saturating this bound is the heat fluctuations. Now, I'm not going to tell you about the proof. What I want to do is see how we can use these kind of general results to gain insight into other physical problems. Okay? So I'm going to give you an example out of Udo Seifert's group. I want to go back to that idea about the heat engine. We're not going to look at a cyclic heat engine. Those are what I'll say is non-autonomous engines where you're driving the system with a cyclic protocol. I could also imagine just coupling my heat engine to a hot and cold reservoir simultaneously and just let it run. Okay, that's an autonomous heat engine. And so you've got a constant flow of heat from the hot reservoir, a constant delivery of work or power, and a constant flow of heat into the cold reservoir. Okay, so we're great thermodynamicists. So the quantity of interest is going to be the efficiency, which is the ratio of power to the heat into the system. And the question is, what is the best efficiency we can do? Well, here the efficiency is, of course, bounded by the Carnot efficiency, 1 minus the ratio of temperatures. We also know from the second law tells us that we can reach the Carnot efficiency if we go really slowly. So if we go slow, then our power is going to go to 0, right? So the second law tells us that one way to get to the Carnot efficiency is to send the power to 0. It's not a great heat engine, but it's very efficient. At the time of this, there was a bit of discussion in the literature because there were some models that had suggested that you could reach the Carnot efficiency at finite power, okay? And so we can try to study this from the point of view of this thermodynamic uncertainty relation. We can choose the integrated current to be our power, our work done over a finite time. And you rejigger the uncertainty relation around a bit, and you find that the power times the efficiency over the Carnot efficiency minus the efficiency times 1 over the power is bounded by a half, a finite number. So this is interesting, because now you can ask the question, let's imagine I try to drive my system to a Carnot efficiency. Okay, I'm going to try to drive my system to the Carnot efficiency. So this thing on the bottom, this NC minus eta, is going to blow up. If I keep the power finite, if I try to do this at finite power, the whole thing has to be bounded by a half. And the only way I can do that is to drive the variance in the power fluctuations to infinity. Right? So it is possible to have a heat engine that works at finite power at the Carnot efficiency. However, the cost is that your power fluctuations are infinite. You are as likely to get work out of the system as put work into the system. Right? So still not a great motor, but at least it resolves this issue. Another interesting example out of Seifert's group is on the efficiency of molecular motors. Right? So a molecular motor, if you've never seen them, they're molecules inside cells. They're responsible for transporting cargo from one part of the cell to the other. They have two little feet and legs. They literally walk leg over leg across scaffolding inside the cell. And one of the kind of classic ways that this is studied is we take our little molecular motor on the scaffolding. We supply some chemical energy in the form of ATP to watch it run along. And then we chemically connect it to a plastic bead, which we laser trap. And as the molecule is running along, we pull back on the laser trap and we see how fast it's going. Okay? So we're good thermodynamicists. What are we interested in? We're interested in the efficiency. The efficiency is the power against the external force over the chemical work. Again, from the second law, what is the best efficiency you can get? What is the Carnot efficiency? In this case, the Carnot efficiency is just 1. This is a work-to-work converter. You're turning one form of work, chemical work, into another form of work, mechanical work. And the second law says you can do that with 100% efficiency. You only get these weird bounds with temperatures when you're doing thermal energy to mechanical energy. Okay, we can now ask, what about the fluctuations of the distance traveled of this molecular motor? We can put it into our bound. We can rejigger it around, and we find that the thermodynamic efficiency is now always bounded by 1 over 1 plus the velocity, the observation time, over the fluctuations in velocity times the force. Okay, there's a lot going on there, but what do we learn that's important? On the right-hand side is purely kinematic information, right? Not thermodynamic information. It's just fluctuations in the distance. What this is telling us is that we can learn about the thermodynamics, the energetics of this system, by just looking at its fluctuations in distance traveled in velocity, okay? This is surprising and nice because actually the data from this to do this was around in the late 80s, early 90s. And you can actually get bounds on the efficiency. Now, this is all to say it is in principle possible to measure the thermodynamic efficiency itself without getting a bound, but you need to be able to monitor at a single molecule level the rate of ATP consumption, the rate at which it's converting ATP into energy, which is in principle possible but challenging. Okay, I'm going to switch gears a bit. I got our plot here. We got an integrated current as a function of time. And so far, what we've been doing is making a vertical slice in this plot. We've been fixing an observation time and looking at the histogram of fluctuations of all these currents, which has some variance. Now, I can also imagine a different problem, a different question, where I make a horizontal cut in this plot, and I ask, what's the first time I reach a particular level of my current? What's the first time that some event occurs? And these first passage times will also have some fluctuations. Now, not surprisingly, these things are related. If anyone has actually tried to calculate these things, in general, they are very complicated related. There are no simple formulas that connect them. However, we were able to show that in a large time, large current limit, these fluctuations are actually just proportional to each other. Which means we can just import everything we've learned so far for first passage time fluctuations. And we can get at kind of first passage times for clocks, that the variance in the first passage time over the mean is universally bounded by 2 over the heat dissipation. So this is very interesting implications for our molecular clock model. Remember, we have this molecule that goes around in circles. And now imagine we had a bunch of these, like 1,000 of them. We started them all at the top at the same time. And we let them run. And every time they complete a circle, we put a little dot here, right? So this guy completed here. This guy completed here. And initially, they all start together. Sometime later, one frequency time, they complete again, but it's noisy. A little later, they complete again, even more noisy, even more noisy. You can see this cloud starts to spread out. And this spread of the cloud is exactly the variance in first passage times that we're measuring, and is exactly capturing this idea that we're losing precision, right? If it was very precise, these lines would march along together with a fixed frequency. So we started to ask, we have these clock things. Can we do this kind of realistic models? There are many models of more realistic biochemical clocks. They're much more complicated than this little thing going around. There are many molecules. They're at higher copy numbers. It's a high-dimensional space. But if you project those dynamics down onto a two-state space, you actually get a cycle. Things just spin around. You can imagine to make a clock in whatever space you're working in. Things just have to go around in a circle. And so you can now ask about what this first passage time fluctuations are going around this circle. We didn't exactly look at that. We looked at the kind of number of coherent oscillations. It's just the precision upside down. And we plotted that against our bound. So in the bottom right corner is kind of this measure of clock precision, dissipation, a function of clock precision. The colored lines are our realistic models, and our bound is way up here in the corner, right? So basically what this is telling us is that this is a fundamental bound on clock precision. However, biology probably has a lot of other things going on besides optimizing the precision for dissipation, right? They might have to be precise, they might have to adjust to perturbations. There's all kinds of things we would like to incorporate over time into these kinds of bounds to really understand the optimal conditions. So with that, I'm going to summarize like the first half of the talk. I've talked to you a little bit about this uncertainty relation, this trade-off between precision and dissipation. Since the original work, there's been a lot of expansions. You can do it with diffusion processes, discrete time. There are tighter bounds that include stuff about the state space topology. There are other derivations that are a million. And there's been a lot of work recently now, the 2025s, on like clock precision, tightening these clock precision kind of questions. So if this is the kind of thing you're interested, my colleague Todd and I wrote a review a few years back in Nature Physics. It is admittedly outdated. The field's moving too fast. It's a few years old. These references are completely outdated. But at least you can kind of get a sense of the field and see where things are going from there. Okay. With that, I'm going to switch gears and come to this idea of response or sensitivity. So there are many situations in physics and biophysics where we have some input, maybe a chemical input, or you apply an electric field, or you apply an external force, and you look at some output, maybe a chemical output, or the motion, or charge distribution. And we look at how systems respond to perturbations. It's a classic way in which we characterize physical systems, right? Response coefficients, mobilities, dielectric constants, isothermal compressibilities. The idea is that if we drive a system out of equilibrium, maybe we can make these systems more sensitive, and can we make that intuition concrete? Now, the motivation for me got started in actually reading a biology textbook, a classic first-year freshman textbook, and learning that there are many biological situations, assays, where it's natural to think about the response of a system, okay? So the first one I'm going to start about here is on the left. This is from bacterial chemotaxis. So bacteria like E. coli have little motors on the end that attach to flagella, long polymers, right, that spin. And when the motors spin, the flagella spin, and it starts zipping along and starts moving. And randomly and every so often, that motor will start to spin in the other direction. The flagella will come apart in random directions, and the bacteria will change directions randomly. And the bacteria will manipulate that bias between clockwise and counterclockwise in order to actually move up concentration gradients in food. It's how it actually finds food, essentially, okay? So you can take that motor out. You can do this nice experiment where you vary the signaling molecule that causes that bias, QYP, it doesn't matter, and you look at that clockwise bias, and you see this remarkably steep switch-like behavior. There's a very sensitive response. Nothing's happening, nothing's happening, nothing's happening, and then a very precise value, the thing switches to the other direction. So you start with low signaling molecule with a counterclockwise bias. You're almost always going counterclockwise. And then there's a switch to a high clockwise probability. This has the form of kind of a Fermi function or a Hill function, right? This is actually the structure. If you are an expert, the Hill coefficient here is 21, which means that's really, really steep, okay? So we think this switch-like behavior, this sharp change, is important for function, okay? Another example here is from the cell cycle oscillator. So, cells are developing, they're replicating. They have to keep time. They have to know when are they supposed to split. What are they supposed to do next? So there's a collection of molecules that go through reactions that oscillate. And what we're looking at here is kind of one molecule versus another molecule's concentration. And again, you see this kind of switch-like behavior, right? And the important thing for us here is that it's a nonlinearity. So if you want to create an oscillator, you need some nonlinearity. And you can do that by burning energy. The last example I'll draw is from gene regulation. This is an image from one of Ernan's papers. So, cells have to express genes. They have to turn that on and off. They do that by varying some concentration of a molecule or transcription factor. What you're looking at here is basically how much the rate of mRNA production, how fast the gene is working, as a function of really a proxy for that concentration. Again, you see this kind of switch-like behavior where you're making mRNA on the left and you're not making it on the right, okay? And what's interesting about all these examples is we believe that they're all out of equilibrium. So traditionally, when people did these kinds of assays in equilibrium, I knew what to do. I'd write down the Boltzmann distribution, I'd turn the crank, I'd do free energies, blah, 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 right? I get what the answer is. I don't have access to those tools. So what can we do now? So before doing that, what I want to take a step back and see what kind of predictions can we get from equilibrium thermodynamics, equilibrium statistical physics, so we can know what kind of a non-equilibrium thing we can do. And so I've drawn here is an example from biochemistry where equilibrium statistical physics was super important to the development of biochemistry. Okay? So what I'm showing you here on the left is a picture of hemoglobin. So hemoglobin, right, is the molecule that's inside our red blood cells. It's responsible for transporting oxygen from our lungs to the rest of our body. The way it does that, it is a tetramer. It is 4 identical pieces, and each piece has a little oxygen binding site, right? So basically, when oxygen gets close to this guy, it will latch on and catch a ride to wherever it's going to go. So the classic kind of assay that one would do is you'd put all the hemoglobin in a test tube, right? And then you'd vary the amount of oxygen in its environment. So when the oxygen concentration is low, what we're going to look at is the fraction of bound sites, right? So how many sites actually have oxygen versus not. So when there's no oxygen in the environment, no oxygen is bound to the hemoglobin, so the fraction of bound sites is very low. As we increase the oxygen concentration, there's going to be a switch, and then when you have more oxygen in the environment, it starts hitting the hemoglobin, right? And it tends to get stuck more, right? So the fraction of bound oxygen is going to be higher. And so again, you get this kind of switch-like behavior. And we think that this switch-like behavior is important for its function, right? The basic idea is these high concentration, high pressure areas are inside your lungs. So as the red blood cells go by your lungs, they're going to grab up that oxygen, right? And then it's going to start moving through the rest of your body. And when it gets to a low pressure area, it's going to dump that oxygen because you need it, right? However, we don't want what we want here is a very steep slope because we want to get to a region where we know we need the oxygen and just dump it all right there. We don't want to trickle it out along the way in regions we don't need it. So the system needs to create a very high, steep slope. So this was originally a mystery in biochemistry. And the way one solves it is actually build a statistical physics model. So I'm going to do that with you kind of briefly, a theorist model, quite generally for this idea of like small molecule binding to a big macromolecule. So if you need an example, think about that hemoglobin. So here's my theorist picture. I have a macromolecule. I have these small ligand molecules or oxygens that are binding and unbinding. And if you build a statistical physics model, right, you have to write down the free energies. The free energy of binding will depend on the external concentration of your molecule. So now what you can do is you write down the Boltzmann distribution and you can calculate what are the fraction of bound sites. It's like doing an icing model with n sites instead of infinite sites. When you do that, you find when the concentration is low, you tend to be empty. When the concentration is high, you tend to be full. And you create this steep kind of switch. And we want to ask, how do you make it very steep? Well, what I mean by that is when I change the input, I get a big change in the output. I get a big derivative, right? And as a statistical physicist, I know how to do that. I know linear response theory. That's all we're really doing. We're looking at the linear response of some output to some input. And so the question then becomes, how big can you make the linear response? In historical reasons, we're going to look at the derivative of the log of the fraction because it's dimensionless. And so you can write this down. What people did is they wrote down Boltzmann distributions. They calculated, they tuned parameters, they looked at other models, and they found that there was an upper limit. You can actually be a little slicker. You can use the fluctuation-dissipation theorem. But what you always find is that this sensitivity is bounded by the number of binding sites n. There is a structural limit coming from the shape of the molecule that tells you how sensitive you can be. This turned out to be really important. One of the reasons it turned out to be really important is you can now do this assay. You actually go and vary the concentration and look at the binding fraction and then try to extract the sensitivity. What you usually do is fit it to a Fermi function if you're in physics. And if you're in biochemistry, you fit it to a Hill function. And if you're in math, you fit it to a sigmoid function. They're all the same function. Okay? But I don't know where everyone's from, so I want to be sure I named one for every discipline. Right? And you can extract the slope of this curve. Okay? And you should get n, the number of binding sites. You can then go to your structure buddies. In my department, they're just down the hall. And ask, do I have n binding sites? Yes or no? And then try to understand what's going on with that scenario. Okay? So this discovery was really important. And with it, you can start to ask, how do I actually saturate that bound? How do I make things really sensitive? Because understanding this notion turns out to be important for biochemistry. And to do that, I'm going to make a new picture. I'm going to try to build another language with you guys about how we're thinking about what's going on. So we started with this kind of static picture, things are bound. right? There's some fraction of things bound, the enumerated states. But really, it's a molecular scale thing, and there are fluctuations. And these ligand molecules, these oxygen molecules, are binding and unbinding, binding and unbinding, binding and unbinding. And all we're really calculating is the average occupancy. So we can create kind of a dynamical picture by listing all the microscopic configurations. Here, if there's just two states, there's empty, full, left full, right full. And I can organize them into a little graph where the nodes of that graph are these states. And now imagine that the system is actually making random transitions between these different states, right? I could start empty, and a moment later the guy on the left could bind, and a moment later the guy on the right could bind, and a moment later the guy on the right could unbind, and there's fluctuations between these different states. And so I can symbolize that by drawing arrows, right? I'm saying that if there's an arrow going up, there's a potential transition between these two configurations where the right guy here unbinds, okay? If there's an arrow going horizontally here, there's a potential transition where the left guy binds, okay? And I've also, for convenience, this will be important later, I've highlighted in purple those rates that depend on the concentration of the ligand molecules, right? So the binding rate, the rate at which I bind an oxygen or a ligand, depends on how much is in the environment. If there are a lot of ligands in the environment, I will bind faster. It's easier. They're bumping into it more often, and so one of them is going to stick. If there are less, it will happen less often, okay? And so within this picture, we can answer that question, how do we create a very sensitive switch in equilibrium? It turns out it's through a mechanism called cooperativity. The basic idea is the binding of one molecule interacts with the binding of the other guy, okay? So in this picture, it's a lot like an Ising model where you have two spins, bound or unbound, left or right, and they're talking to each other, okay? And as long as they talk to each other, I can get really strong sensitivity, just like in the Ising model. I can get really big switches from up spins to down spins as long as they talk to each other, especially near a phase transition. And this is exactly the same mechanism. Here, I'm going to give you a dynamical picture. Basically, what you want to do to make this happen is I want to destabilize some states and stabilize others by varying the rates. I want to send some to be small by making them, which I make blue and thin. And I want to send some to be fast, which I make fat and red. And if I do this, I can make a really great switch, right? So here, I've stabilized the guy on the left. I've made the out rates in blue small, so I get stuck there. The guy in the corner is destabilized. The out rates here in red are very big, right? So what happens basically is I'm stuck in the guy in the top left. Occasionally, I jump, I bind, and then I quickly exit one of the two spots. So I'm never stuck in that half-filled state, which means I'm spending all my time empty and all my time full. I'm switching back and forth those two extremes. And then as I vary the oxygen concentration, I'm biasing those two. Empty, 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 full, right? And that's the cooperative mechanism dynamically, and that's how we create this kind of very high sensitivity in equilibrium, okay? This was really important back in the day. But life is out of equilibrium. I want to do the same thing. I want to tell you the same story. I mean, this was a really successful story. Can I do the same thing, but now imagine I'm driving the system out of equilibrium somehow, okay? There's energy consumption, there are non-conservative forces, I don't know, right? I want to be kind of general and then see what that means. So what we found is that instead of the number of binding sites, which is a structural quantity, we found that in a non-equilibrium system, there's a topological quantity in this state space that gives us the ultimate bound. The basic idea is I'm going to go through my system of states. For every state, I'm going to color it green if one of the outgoing edges is controlled by that ligand concentration, if I'm perturbing it, right? So remember those purple guys depend on the thing we're controlling, the oxygen concentration. And so this guy in the top left corner has two purple guys going out. I'll color it in green, okay? This guy in the top right corner has one purple line coming out. I'll color it in green. But this guy in the bottom right corner, which is the completely full state, has two out rates, exit rates, that don't depend on the concentration of the ligands. I'll leave it black. And the idea is what we're going to do is color these guys, count all these green dots, which we call the support of the perturbation. And the sensitivity is universally bounded by the number of green dots. So all you have to do is build your model, identify which kinetic rates depend on the thing you're controlling, count your green dots, and I can tell you how sensitive the system can be for any model, any discrete state Markov process. Okay, so we can prove this. We have a theorem, fun stuff. Let's talk about applications instead, all right? And what I want to do is I want to go back to this gene transcription we talked about at the beginning. Let's build a model of gene transcription. So here's my theorist depiction. So what is a gene? A gene is just a region of DNA. There's a part that encodes for the mRNA over here. And before that region, there's a long part of DNA with binding sites for these transcription factors. And these transcription factors bind and unbind from the DNA. And they tell the cell when to start making mRNA, right? When do we turn on? When do we turn off? So in this picture, they're just binding and unbinding, and you have to pick a model rule. A typical rule is, let's say, when all these sites are bound, the gene turns on, you start making mRNA, right? We know this is all good for the vaccines and things. And the question is, how sensitive are we to this concentration of ligand? Now, so if this was an equilibrium model, I told you that the sensitivity was bounded by n, the number of binding sites. Okay? It turns out that in bacteria, this is a very good model. Equilibrium binding works really well. It agrees with the experiment. However, in eukaryotes, things with nuclei, there's some evidence that the equilibrium binding models aren't as good or don't work. And people have suggested that there may be a non-equilibrium mechanism at work. Okay? The question then becomes, what is the highest sensitivity you could get in this case? Well, you color your green dots in your model, and you find that there's an exponential increase in the sensitivity. Instead of going like n, it goes like 2 to the n minus 1. And so you can get way better. The way I like to think about this, again, is kind of an uncertainty relation kind of trick, right? Well, really a trade-off in resources. So I want to create a really sensitive switch. It's important that I turn on at a precise concentration and not kind of dribble on and off, because that would screw with the function of the cell. Okay? So what's the trade-off? Well, I could be really sensitive and not burn energy, be at equilibrium. Then I need to create a lot of binding sites. So I have to invest resources in making a long piece of DNA with lots of binding sites. However, I could do it with less binding sites if I'm willing to burn energy. You can do it exponentially better. So you invest energy, or you invest the materials of creating those binding sites. Those are your two choices. Now, we weren't the first people to study this problem, right? It was originally studied computationally. And despite using relatively, what they did, they sampled a bunch of rates, and they looked through a bunch of models, and they tried to figure out what the maximum sensitivity was. And despite rather sophisticated optimization programs, they did not observe this sensitivity. It turns out to be really hard computationally to identify these things because they're very extreme. Okay? However, we have a theorem and a proof, so we can show that it is in fact true, and this is the best you can do. Moreover, we can give you the conditions for equality, and this led us to identify a novel biochemical mechanism for sensitivity, which we call nested hysteresis. I'm not going to go into detail, but very high level. Basically, there are two ingredients. There's a hierarchy of timescales. For every binding site, you have a different timescale of binding. Moreover, there's a binding and unbinding in order, but in opposite orders. You bind in one order and unbind in another order. Okay. Let me flip to another example. Again, I want to go back to this bacterial flagella motor. This is a nice example. I'm showing a picture up here in the top right. I don't know what it is, but it looks cool. On the bottom, again, is that clockwise bias as a function of the concentration, right? We got this steep switch-like behavior, okay? And the question is, what is the origin of this switch-like behavior? So you can go into this motor, and you can look at the structure, and you find that there are, in fact, 34 binding sites for this signaling molecule, okay? You can then look at this assay where you do this switch, and you find that the steepness, or Hill coefficient, is actually 21. So within our equilibrium framework, this is fine, right? I needed more binding sites than my sensitivity. 34 is bigger than 21. I should be cool, right? Equilibrium modeling should be fine. However, other experiments have shown that all those 34 binding sites are never all bound at the same time. Somewhere between, it varies between 2 and 13, right? A difference of 11. So if only 11 binding sites are being used, I don't have that 21 that I need to get to the sensitivity. So how do you reconcile these two things? Well, U-High-2 provided a model, a kinetic model for us. And the basic idea is that there are two kinds of states, clockwise states and counterclockwise states. And each state is marked by whether it's counterclockwise or clockwise and the number of bound signaling molecules. The details aren't so important. You can color the green dots. You find that this model, the sensitivity, scales with 2 to the number of binding sites. And for that span of n binding sites, we get 22. suspiciously close to the 21, right? So what we're saying here is that our theory is saying that these kind of non-equilibrium models are indeed sufficient to explain this phenomenology, okay? So with that, I'm going to try to sum up. I tried to argue that we can really make serious quantitative statements about kind of energetic trade-offs between sensitivity and structure, topological, energetic properties of non-equilibrium systems, much of the way we do in equilibrium thermodynamics. We've applied this to biochemical switches, sensing, kinetic proofreading, gene transcription, flagellar motors, so we can talk about a lot of different things. What I've talked to you about today is really the coarsest result. We have a number of results that are tighter, that include more information about the topology of the state space. We try to look at diffusion processes. We try to look at chemical reaction networks. Turns out that they are significantly more complicated, and we've made partial directions in that, but I can't really tell you a clean story. What I've hopefully convinced you of this idea is that we really have the tools now to make these intuitive ideas that providing energy to a system, driving it out of equilibrium, can create a new function, and we can quantify that, understand it by deriving these kind of inequalities or trade-offs to understand the limits and optimality conditions that delimit the space of non-equilibrium behavior. So thanks very much.
Okay, we have time for a few questions. There.
Thank you very much for the great talk. I have a question about the mean first passage time. If memory serves, you said it's not in a long time, high current limit.
Mm-hmm.
But I was wondering, how do you know that you're on this limit without running the process and seeing if it's
So how do I know if I'm in the large current limit, like experimentally or theoretically?
even in computation, like if you set up some unknown, previously unknown model, you don't really know how things, like what's just the internal scale. How do I know if I should rely on this?
So if I don't know so there has to be kind of a longest scale that I'm aware of. Formally, what we're working in is in the large deviation limit, or the thermodynamic limit. So really, you should be working in that scaling regime. And I don't have any obvious way to know a priori without just seeing if you get the right scaling on the fluctuations. Did that answer your question?
Yep, I think so.
Okay, thanks.
David?
Yeah, I wanted to ask about the sensitivity bound. And I wanted to try to get some more intuition as to where the exponential scaling comes from. So in equilibrium, I understand that you maximize that inequality when you maximize the variance, the fluctuations, which you get about through having lots of correlations amongst the sites. So that's where you get quantitative that comes directly from interaction. Out of equilibrium, can I think about it as still maximizing the correlations amongst the states that I'm transmitting through the dynamic evolve? Or is it really something that comes about from a breaking of a fluctuation-dissipation relationship such that the response is not related to the variance?
Okay, so what is the origin of the exponential scaling of this bound? Is it related to fluctuations?
Is it because the fluctuation-dissipation relationship is broken? Or is it because I have a new mechanism correlating sites so that my variance can be more?
Is there a new mechanism? I mean, in many respects, this kind of nested hysteresis, one could think of as a new mechanism whereby the system is kind of transitioning between these slow and fast states, and it's binding and unbinding, binding and unbinding, binding and unbinding. And so there's kind of a dynamical correlation in that sense. I have not framed it specifically in that way. To be honest, I've really struggled to create a nice physicsy explanation, in part because the math was so hard. But I hope to get better at that over time as we can explore new examples.
Hi, thank you. Very interesting. I was wondering your results about sensitivity and the function of concentration out of equilibrium. Is there an analogy between that and kinetic proofreading, but now instead of like an energy gap, concentration kind of plays a role of energy?
So is there an analogy between this sensitivity bound and kinetic proofreading? In fact, it's more than an analogy. In fact, the results apply to kinetic proofreading, which is in our paper, actually. And the kinetic proofreading can be framed as kind of a a sensitivity to an energy difference, and then we'll look at the kind of efficacy. And our bounds can recover the known bounds and extend them in some regions. We're hoping to make them even better this time, but yeah. So yeah, there are a number of things that we looked at that can be switches, gene transcription, kinetic proofreading. They all can be kind of framed as, anytime you can frame things as sensitivity, we can help you out.
I was curious about in the sensitivity, about the impact of the actual speed limit of the process. Which is to say, if the fastest thing is diffusion limited, you have to stack this hierarchy of timescales on top of it. Does that mean that inevitably it's going to be slow to react to this time?
So the question is, if I have some sort of cap on my speed, speed's important now, and I still want to create this as sensitivity, can I do it? The short answer is no. Adding speed turns out to be really hard. I tortured an REU student for a summer trying to figure this out. I would love to understand speed. It's really important. I don't know how to do it yet. But maybe you should approve my next grant application, and we'll work on it.
So all this function under Markovian, right? Especially for the transcription part.
Yeah.
Under Markovian.
So the question is, everything's under, is that the question?
Yeah.
Yeah, so everything is under the Markovian assumption. I don't see that as a big limitation because you can always lift a non-Markovian process to a Markovian process by including more states, and you just have to apply the theory there. Yeah.
I actually want to ask you a question, and I know you were trying to spare us all the details of the rate and all of that, but you said that most of the stuff before, like, the sensitivity thing, for example, had been done computationally.
Yeah.
So I'm just curious about how, like, the trick to getting these analytical bounds. You said they're hard, but is there some intuition about, some broad picture of how you get those and what was the key step?
Okay. So a technical question. What was the key step to getting these bounds? There's something magical about these Markov. If you're studying steady states of these Markov chains, there's a result from graph theory that actually means you can write these steady state distributions using pictures, using inherited graphs from these pictures. It's a lot like Feynman diagrams. There's like a Feynman diagram language. And that meant that we could study these problems by manipulating pictures instead of algebra, and that allowed us to kind of make these results, right? One of the things we learned from the numerics, the reason that's very hard to sample, is that you really need to be sampling on a logarithmic scale. And you need to create, like we saw, we need to create these very wide timescale separations. And if you have a lot of parameters and you're sampling them, it's hard to get that by accident. You have to really try for it. But once you know how to do that, you can actually do it numerically pretty easily.
I have a question about the end of the first part. You mentioned that the fact that the bound of the clock was so far away from what actually happens indicates that since the biological system is optimizing something else. Are there cases in which you can look at like a simple biological system and apply multiple bounds and try to get out like what combination of these attributes evolution has decided to
Okay, so the question is, I said that maybe biology was optimizing something else besides this thing, and maybe there's some combination of things we could try to do. I don't want to harp too much on like biology is optimizing things, right? We don't really know that that's happening, but one could put it as a hypothesis. There are biological examples where this is saturated. You can think of equalities as testing for some equality condition. How close are you to the equality condition? It turns out, in this case, it is Gaussian fluctuations. There is a biological example in molecular motors where you have Gaussian fluctuations. If the molecular motor is walking very slowly, the fluctuations are Gaussian. If you grab onto it and grab it so it can't walk, so it's stalled, it turns out those fluctuations are Gaussian as well, and things work there as well. Your broader question about can I combine things, that's definitely been something going on in the field. I showed you one result that really has one constraint. When we did this, there was an explosion. There are many bounds now that constrain other things, more dynamical things that have to do with like how active or how hoppy the system is, how driven the system is, the state space typology, the speeds of things, the first passage times. And people have tried to combine them into a framework to say like, this is the thing that's being optimized. If you email me, I could try to find the papers, but I personally haven't kept up with the literature in that way. But I mean, it's a cool idea.
I have one more question. about what you can do with energy, but you don't give us much of an intuition of the scale of energy necessary to carry out these functions, and at least in the context of biology, we might have some ideas. So, like, well, what's your intuition on that?
And that, so what is my intuition on the scale of energy? So I think it's easiest to answer in the kind of second half of the talk where I actually didn't talk about energy. I just said I was out of equilibrium. So question, how far is far? The kind of energy unit in biology is about 24 kT. That's, as a statistical physicist, I measure everything in thermal units. That turns out to be huge. That turns out to be quite large compared to the numbers that these theorems have. So there is an argument that for many biological processes, you're basically infinitely far from equilibrium. And one can think about it that way. I think it's a fun game to play, though, is to try to experimentally tune that, to like lower the energy supply inside a cell and see what happens and see if you can futz with things like that. But largely, I would say you're very far.
Just, I mean, kind of on that, I was just kind of thinking, I was curious your perspective generally, like, you know, what the field needs in terms of experiments. Like, one can imagine, like, you know, abiotic things with colloids rotating or something, or you could, you know, then, you know, I know you interface with biologists, so as you know, these experiments, it's hard to change just one thing, for example, in cells, and, you know, it's not as, the mapping theory is much harder. So what do you think in general the field needs in terms of experiments to kind of put these into play? So within the like modern non-equilibrium thermodynamics, the experiments that have been most influential have been largely like synthetic experiments, very clean things. So early on, a lot of the tests of these my results, but earlier results were just laser-trapped colloidal beads, right? And I think in this context, with these biological things, I think synthetic biology is really the place to play around with these things, to kind of tune parameters and see if we can move things to that limit. I think there's a lot more, as a theorist, I'm more excited by the other direction, where what are the realistic constraints we need to put into these systems? Is it energy? Is it topological structure? Is it binding motifs? How do we incorporate those things to really fill out this phenomenological space and kind of have that dialogue? But yeah, synthetic biology.
More questions? Jordan again?